Question number 121. Mark the below given statements as true or false and select the correct option. A. DNA is better material for transmission of genetic information than RNA, hence preferred as genetic material. No, RNA is better material for transmission of genetic information than DNA. So, this is incorrect. B statement, absence of free 2' OH in deoxyribose sugar of DNA makes it more stable. It is true, because if you see the sugar of the RNA, you will find that 2' OH as well as 3' OH both are free in the ribose sugar of RNA, and the deoxyribose sugar has only 3' OH, not this 2' OH. So 2' OH is not present in the deoxyribose sugar, and this absence of free 2' OH in the DNA makes it more stable. It is true. DNA has ability to generate its replica. It is also true. So false, true, true. So the correct option for the given question is option number 3. Let's move to the next question. Question number 122. DNA polymerase is known to perform all of the following functions except removal of RNA primer is done by DNA polymerase 1. Synthesis of Okazaki fragments. Yes, because synthesis of Okazaki fragments is done by DNA polymerase 3. Now third, DNA synthesis in 5' to 3' direction is done by the DNA polymerase. Fourth, joining of discontinuously synthesized DNA fragments. Discontinuously synthesized DNA fragments are the Okazaki fragments, and joining of these fragments is done by the DNA ligase, not by the DNA polymerase. So this is the function of DNA ligase. So the correct option for the given question is option number 4. Let's move to the next question. Question number 123. Given below is the base sequences of known coding strand of a transcription unit of DNA. How many amino acids would be coded by the mRNA formed by this given transcription unit? This is the known coding strand. Means you have to make the complementary strand of it, because this is known coding strand. It is the template strand. So make the complementary strand, or you can make the mRNA from it, because this is a template strand for the mRNA. Now it will be for T, it will be A. For A, it will be U, G, U, 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 C, C, U, U, G, G, U, G, A. Now, you know that a triplet behaves as a codon. So, first amino acid, second amino acid, from here, third amino acid, UGG, fourth amino acid, UGA will be a stop codon. This won't code for any amino acid. So the total number of amino acids will be formed are 4. So the correct option for the given question is option number 1. Let's move to the next question. Question number 124. Choose the phenomenon which occur in cytoplasm of eukaryotic cell. Splicing, tailing of hnRNA, charging of tRNA, capping of hnRNA. The hnRNA formed in eukaryotes is processed, and the process is splicing, tailing and capping, and these processes take place in the nucleus of the eukaryotes. So splicing, tailing and capping, these processes takes place in the nucleus of the eukaryotic cells. Charging of tRNA, it's a step of the translation or the protein synthesis, and this step takes place in the cytoplasm of the eukaryotic cells as well as prokaryotic cells. So the correct option for the given question is option number 3. Let's move to the next question. Question number 125. Which of the following RNA polymerase are involved in rRNA synthesis in eukaryotes. The major rRNA is synthesized by RNA pol 1, but 5S RNA is synthesized by RNA pol 3. So both RNA pol 1 and pol 3 are involved in the synthesis of ribosomal RNA. So the correct option for the given question is option number 3. What about the RNA pol 2? RNA pol 2 is responsible for the mRNA synthesis. Let's move to the next question.